Hey, welcome back to part 2 of chapter 6. When we talk about the structure of long bones, and these are bones that are, we call them long because they're very long. There's no other kind of way to describe them. Like your femur is a prime example of that. Fibia, tibia, humerus, and so forth. But really the femur is the classic example of this. They have common structures associated with them. Diaphysis, it's the shaft of the bone surrounding the medullary cavity, and this is that central cavity that we think of where the bone marrow resides, which is surrounded by that cortical bone structure. So that medullary cavity is home to the red blood marrow during child development, but in adults that are replaced by yellow bone marrow, except for proximal epiphysis of the arm and thigh bones, but otherwise it goes from yellow to red, but basically saying the yellow marrow has more adipocytes in them and the red marrow has more traditional red cells, blood cells in them. They have epiphysis, which is at the ends of the bones, either proximal or distal. They're more spongy, so they're trabecular bone because of that. These are the ones where we find that hyaline and cartilage covering them to form those articulating joints, so articular cartilage. There's then a growth plate called the epiphyseal plate. Traditionally, this is a hyaline and cartilage initiates there and grows, and then when you stop growing, that hyaline and cartilage just goes away, forming that calcified growth plate. That's what that really is. So because of that, we have a epiphyseal line. This is where ossified bone stops growing and where we find more of that cartilage-like tissue. Just surrounding the outside of the bones, there's this connective tissue called periosteum, which provides bones with protection, housing for osteoblasts, osteoclasts, our stem cells for those, as well as just the blood vessels and supporting collagen and protein structures to support the bones themselves. So it's not considered it's not the bone themselves, but it's a connective tissue surrounding them, providing them proper functioning, as well as strengthening for the tendons and ligaments inserting into the bones. So here's our periosteum, right, just surrounding them with a fibrous protein layer. Then we have our actual bone matrix here. So we have this other layer called the endosteum, which is a single cell layer of connective tissue that lines the internal spaces of the bone. As we can zoom in here, just right there. This is where we'll find those osteoblasts, clasts, and stem cells. During my doctoral work, I worked with a coworker who actually took stem cells out of mice long bones, and we were looking for these endosteum cells because that's where they're going to reside, that single layer just inside the cavity, the medullary cavity there. And when we're looking at the bone overall, here's our diaphysis, right, the shaft, right, or the body, right, from the other terms that we learned here. Then we have the proximal epiphysis and distal epiphysis, and those are going to have more trabecular bone located in them because, they need to withstand those jumping forces that will transfer the bone, the jumping forces into the core of the bone. Then you have your red marrow inside a trabecular bone in the epiphysis versus the yellow bone marrow more in the medullary cavity. Right. So we have different structures there. When we're talking about the physiology of them, when we look at the differences between the flat bones or short bones and irregular bones, flat bones don't have any diaphyses, right, or epiphyses, right? There's just spongy trabecular bone compacted between cortical bone, right? So no distinct regions there. When we look at short and irregular bones, they tend to be also similar to flat bones, that they have compact bone that surrounds spongy trabecular bone, but they do have more epiphysis-like aspects of the long bone, but they don't have any diaphyses, right? So they don't have any core trait to them like a medullary cavity, but they're long in nature, but the core tends to be more just trabecular epiphysis, like core trabecular bone with a cortical bone outlining them. Now, when we look at bone development here, there is a number of steps to them that we have to keep track of. There's intramembranous ossification, so this is where embryo tissue, specifically connective tissue, starts to ossify, get that bone matrix that we think of. Then we have endochondrial ossification, where we actually start ossifying specifically cartilage and not just early pre-development connective tissue. So these two different types of ossification are still ossification in the sense that we're taking like cartilage soft tissue and adding more stiff hydroxyapatite crystals and just general bone mineralization. But what we're trying to do here is we're trying to remodel the woven bone into the lamella bone. Then after that, right, then we can start ossifying, and this is where the differences start to be distinguished. So in endochondrial ossification, this is where we're forming bones like in the skull, the mandible, epiphyses of the clavicle, and so forth. It starts in the 4th week of development, so very early on. And then we have ossification of that cartilage from the 4th week, starting at week 8. But this tissue doesn't truly begin to ossify until you're about 18 to 20 years of age. So this is what we traditionally think of those growth plates ossifying and growth stops. This is what we're really referencing here. And there's 5 stages of this ossification, looking at stages based on age, but it starts early off in the womb. So endochondrial ossification, really ossification in the sense that it starts becoming calcified with bone hydroxyapatite crystals, week 9. So we have that pericardium starting to form, and then most of the bone is just cartilage at this moment, right? Then we have later on, we have starting ossification here. But really one thing I want to note is that the stem cells, mesenchymal stem cells, are the ones forming the bone. And these are the ones that make the bone cells, right? So this early week 9 development is really just cartilage tissue surrounded by a pericardium, and that's it, right? When we move on from week 9 to month 3 in the womb, you really start to have blood vessels going in the pericardium. You start to have true osteoblasts form inside of there, and therefore you start to have a bone collar formation where you have the calcium formation, hydroxyapatite crystals created, and so forth. Additionally, you have those chondrocytes that initially made the cartilage dying and or leaving, right? This leaves the open spaces for them, the lacunae from which those osteoblasts can become osteocytes. Then from week 3 on through adolescence, we have what's called the primary ossification, where osteoblasts and osteocytes migrate to produce proper calcified bone, as well as proper ossification centers, kind of like where you see that bone marrow here. Then you have blood vessels, that grow into large lacunae in the cartilage tissue, and And then osteoclasts can remove bone if they need to, really from the center diaphysis to form that medullary cavity, right? So this is where osteoclast activity is an example of where it's good. Moving on to secondary ossification, this is where inside of your epiphyses by osteoblasts, you start having those epiphyses where you have the trabecular bone occurring, but really this is more woven bone at the moment. You have a secondary ossification, such as in the heads of the bone themselves, right? Because we have the primary down in the diaphysis region and the secondary in the epiphysis regions. The key here is they don't enlarge, right? Secondary ossifications do not form a medullary cavity, right? Because they're forming trabecular bone that don't have that. So the primary ossification centers that form that medullary cavity happen early on, and then the secondary happen. So that's why we have a primary and secondary aspects occurring with them. All of this is occurring on really through adolescence. Obviously, you need most bone formed before birth, but there's still a lot of ossification occurring, right? Because when we talked about the number of bones in the body, between an infant and an adult, right, you decrease the amount of bones you have because of this kind of ossification factor. Now, inside the adult mature bone, you're done growing. You don't have any cartilage from which you have that growth plate, which we can kind of see in part 4 here. You have a distinct clear region of a trabecular spongy bone and a compact cortical bone, and they're fully developed. You see that epiphyseal plate, which then becomes the epiphyseal line here, right? Because this is the growth plate, right? The top part is cartilage, and then it ossifies down below, right? So this is what an adult looks like. Articular cartilage, epiphyseal line, and then trabecular and cortical bone. So when we talk about bone growth, we were talking about bone development. There's things that occur here that combine both kind of cartilage tissue and bone tissue. And all of this is occurring at the epiphyseal plate, which then becomes a line there, but it has 5 distinct zones. Really, you form new cartilage first, which is called interstitial cartilage because it's inside, right? And then you have apositional growth alongside of the plate to form on top of that surface of that cartilage to calcify the bone. So this epiphyseal plate, we'll have an image coming up, becomes a line. Really, this stops all growth, and when that line is formed, depending upon the bone, it happens between 12 and 25 years of age. So most of you are probably still growing in some of your bones to some degree or fashion. When we have just articular cartilage there, that does not ossify and that persists through life. That's where your joints are. You don't want this to ossify. The apositional growth really is occurring because it's occurring in opposite directions, right? So here's are the 5 zones. We have a resting cartilage zone where we have our chondrocytes. Then we have that are not really dividing, very slowly, if anything. Then we have proliferation where new cartilage is being produced. This is on the epiphyseal side of the plate where the chondrocytes are dividing. Then you have hypertrophy where the chondrocytes are getting large and mature. Then a zone of ossification or calcification where the bone matrix is turning in the cartilage matrix is turning into more true proper bone material. Chondrocytes die, and then your osteoclasts are there on the diaphyseal side, right? So towards the medullary cavity side, replacing the cartilage. So here's what I'm talking about here, where you have your epiphysis, right? Your plate, epiphyseal plate here, and then you have your secondary ossification center and primary. So on the diaphysis side, right, you have ossification zone and that turns into proper bone there. So here's our zones, right? So we have our resting cartilage zone. Then we have our zone of proliferation where our chondrocytes start stacking up and growing, pushing up or out as you could see it. Then you have your zone of hypertrophy where these chondrocytes enlarge. Then the fourth zone where those chondrocytes start to die, osteoblasts come in, and then you start having ossified bone here. So what's happening is you're really first growing cartilage up and away from the medullary diaphysis side, and then the back side that's lagging turns into the bone. So the epiphysis increases in size, and this also occurs to the articular cartilage part. It happens also in bones with no epiphysis. It does happen in short bone. It's just slightly different. It's just the chondrocytes at the surface tend to act more like the resting zone. And this also is true in when you're forming the articular cartilage. They also have this resting zone. When the epiphysis reach their full size, like as an adult, the growth of the cartilage is done and the replacement of the bone occurs. And then articular cartilage occurs throughout life and does not ossify. And this is what this bullet point here is also trying to say, right? So when the epiphysis reaches their full size, when that growth happens and stops, on the articular side, the growth of the cartilage is occurring in the joint, but it's not replaced by bone. So when we talk about bone as well, it also increases in width, kind of similarly to up and down, but not truly up in the same way. This occurs throughout life. This doesn't stop. In adults, the diameter increases and decreases, mainly from the fact that the main thing regulating diameter is the osteoblast. There are no cartilage associated with the diameter, so that's the main thing here. And this is from the periosteum, so outside the bone, right? As well as somewhat inside the bone as well, in that endosteum as well. So osteoclasts and sites work in these regions to expand the width as well as decrease. So this is constantly happening throughout life, versus making the bone longer is only a developmental aspect up to a certain age. Now, there are factors that affect bone growth. One of them is, an example here is rickets, which is the lack of vitamin D. There's also the lack of vitamin C, which is scurvy. Right? You need your vitamin C. So what's happening there is the osteoblasts and scurvy are not producing enough collagen correctly. And then in vitamin D, calcium is not being absorbed properly, so you can't lay down proper amount of calcium, so your bones are actually softened, right, from the initial PowerPoint slides early on. So these are two examples. There's more, but these are two why we need vitamin D and C. If you remember from our chapter 2, vitamin D is a fat-soluble vitamin and vitamin C is water-soluble. So these are both vitally important. Other example of things that affect bone growth is obviously hormones, like growth hormone, which really comes from the anterior pituitary gland. And this is stimulating that interstitial cartilage growth, right, at the epiphyseal plate there, creating that apositional bone growth. And then, of course, thyroid hormone that causes growth in all tissues. Reproductive hormones like estrogen and testosterone also affect growth in the sense that they will close those epiphyseal plates, right? So you have this explosion of growth due to these growth hormones and sexual hormones like estrogen and testosterone, but at the same time they stop. So they cause you to grow and then close those epiphyseal plates. Now you can have a thing called gigantism, example, and then dwarfism, insufficient and excessive growth hormones. And this image on the left is an example of two individuals that have either one. So we'll pause the video there. In the next set, we'll talk about like bone remodeling, which occurs throughout life.